Hello everyone, my name is Yusof. I'm a consultant rheumatologist from Leeds, and I'm reporting live in Chicago for the ACR 2025 conference. In this conference, there have been so many abstracts pertaining to cell-based therapy using CAR and also bispecific T-cell engagers. So I usually don't cover early studies, for the audience understanding. But I do find that this abstract really interesting. It's super sexy and super new therapy. That's why I just want to bring this abstract, into the audience. So the abstract that I'm talking about is abstract number 1535. So this abstract is a T-cell engagers, but it is using the pan gamma delta T-cell engagers. So I'm so delighted, to be joined by the co-authors of the abstract. So we got Dr. Kate Rochlin here. Hi, Kate.
Hi, thanks for having me.
Yeah, thanks for joining us as well. So what I just wanted to know, can you please explain about your compound? What make it different and exciting compared to other current T-cell engagers?
That's a great question because there are a lot of T-cell engager approaches out there right now. And what we really love about what we do is that we're leveraging the power specifically of the gamma delta T cells. So this is a subset of T cells that are incredibly cytotoxic, very powerful killing cells, but they don't secrete the cytokines that induce the toxicities that you see with a lot of the current T-cell engager approaches on the market. So what we've actually done is we've built an engager that targets CD19, and it targets the gamma delta T cells using the gamma delta TCR. So we don't target through CD3. That means we don't run the risk of the toxicities that you typically see or the cell-based exhaustion that you can see with other engager approaches. And then importantly, we've tried to overcome the challenge that gamma delta T cells are a very low percentage cell in the immune system. Very powerful, small but mighty. So what we've done is we've developed a gamma delta T cell expansion domain that actually allows those cells to expand in vivo. So we're activating and expanding those cells to be able to drive deep and complete B cell depletion using our engager.
Oh, that's really exciting, and certainly stand out from other T-cell engagers. So in terms of this abstract itself, so you've done some in vitro studies. Can you just briefly summarize what were the key points from the study?
So in our poster, we were able to demonstrate that our engager is in fact a pan gamma delta T cell engager, meaning that we target a region on the gamma delta T cells that hits multiple gamma delta T cell subtypes. So both the V delta 1s and the V delta 2s. So we're able to drive cells that both hone to the tissue as well as the cells that circulate in the lymphatic and blood systems. We've also been able to show that compared to commercial B cell depleters, such as blinatumomab or Mazu, that we can drive complete B cell depletion in vitro comparable to those two depleters. And that's very exciting for us because it tells us that we don't need to activate through CD3 and drive all of the T cells. We can specifically activate and expand the gamma delta T cells, and they are powerful enough to drive complete depletion. So that was very exciting data for us. And importantly, we do it without any elevation of the CRS-inducing cytokines like IL-6 that are so concerning, particularly as you move into that autoimmune space and you really don't have room for those toxicities at all.
Oh, this sounds really exciting. And in terms of where do you go from here? Would you like to explain a bit more?
Exactly. So I think we've been able to show now that we really get nice gamma delta T cell expansion. Importantly, we've shown that by activating both of these immune compartments, V delta 1 and V delta 2, we can actually cover depletion across a spectrum of autoimmune conditions. So in some patients with more advanced disease, you have one compartment that's more responsive than the other. So now we're moving this into animal models. We're looking forward to bringing that data out in the next 12 months or so, and then looking to bring it into the autoimmune space. And so we're just very excited to be here sharing our data and hopefully bringing the power of gamma delta T cells from oncology, which is where we started, into the autoimmune space, where we really think there's room for a powerful cell type with a lower toxicity profile.
Yeah, thank you so much for the wonderful in the summary. And talking about space, I think we definitely will watch this space for this molecule. So I hope you found the interview interesting, and follow me at U6Yusof on X, and also all of my colleagues at Room Now for further content coverage. Thank you, everyone. Bye-bye.
Thank you.